Okay, welcome back. Let's take a look at our concept check questions. So Maria has type O blood, so we know that O for type O, she has to be little I, so both of her parents must have provided her with the little I. Her sister is AB, so she must be big I A, big I B, so she must have got a big I A from one parent and a big I B from the other parent. So the only way that this could happen is if the parents, big I A, little I, and big I B, little I. And in fact, parents with those two blood types could produce children with all four blood types. So type O is possible, type AB is possible, so is type A, and so is type B. How does the sickle cell disease demonstrate the concept of pleiotropy? Remember the list of phenotypes that I said were associated with sickle cell? That's how it exemplifies pleiotropy, the fact that the presence of this single allele leads to all of those phenotypes, not just a single one. If you cloned some mice, would you expect them to be identical as they age? The answer is, of course, no, because even though they all share exactly the same genes, they're going to experience the world a little bit differently. And as we know, epigenetics, the effect of our environment on our genes, changes the expression of those genes. So over time, as they experience the world a little bit differently, they will no longer be identical. It's the identical twin question again. So one of the issues that people confronted when they finally recognized just how significant Mendel's work actually was that there had to be some vector, some vehicle for carrying Mendel's genetics particles or inheritance particles, what we now refer to as genes and alleles. And people very quickly realized that it had to be the chromosomes. The processes of mitosis and meiosis had been figured out in the 1880s and 1890s. And it was quickly recognized that the chromosomes were the only thing that passed from one generation of cells to the other, or to the next. And therefore, it was likely that chromosomes were the only thing that passed from one generation of individuals to the next generation of individuals. So it seemed logical that Mendel's genetics particles were located on chromosomes, and eventually experiments would demonstrate that that was in fact the case. And so we now refer to the physical location of genes on chromosomes as their locus. That's the singular. Loci is the plural. So the locus of a gene, or the locus of a gene, is just its physical position, its physical location on a chromosome. And it's the presence of the genes on the chromosome that actually accounts for Mendel's observations. And we've talked about the law of independent assortment, and it's the fact that the alleles on separate pairs of homologous chromosomes that accounts for the observation that of the 9:3 to 3:1 ratio in the F2 generation. If the alleles were on the same chromosomes, we would see a different pattern of inheritance. And in fact, that was something that became recognized fairly quickly. Within 20 years or so of the recognition of Mendel's work, it became pretty obvious based on what people were seeing that number one, there were far more genes than there were chromosomes. And number two, as a consequence, not all genes could be on separate pairs of homologous chromosomes. Some genes actually had to be on the same chromosome. And the term linkage was coined to identify this. Genes that are said to be linked are genes that are located on the same chromosome. And because they're on the same chromosome, they tend to be inherited together. And for a while, that was the only thing that was understood. Either the genes that you were following were on different pairs of homologous chromosomes, and therefore they followed Mendel's law of independent assortment, or they were located on the same chromosome and were linked and were inherited as a package and did not follow Mendel's law of inheritance or independent assortment. But eventually it was recognized that crossing over also existed. And in fact, it was doing a test cross on a pair of genes to figure out whether they were on the same chromosome or on different chromosomes, because a test cross can actually tell you that showed the existence of crossing over. And so then people had to first accept the crossing over existed, and then secondly, observe it in the microscope. And eventually people were able to observe that crossing over actually does occur. And so now you have three situations. If the genes are on separate homologous chromosomes, or separate pairs of homologous chromosomes, then they're going to follow Mendel's law of independent assortment. If they're on the same chromosome and close together on that chromosome, then they're going to act as if they're linked, because they are linked and they'll be inherited as a package. But if they're far enough apart on the same chromosome, crossing over can then separate them. And if they're far enough apart on the same chromosome, they'll actually be separated by crossing over frequently enough that they'll look as if they're on separate chromosomes, and they'll follow Mendel's law of independent assortment. It's a further twist on this whole question of, are genes on the same chromosomes or are they on different chromosomes? Crossing over adds an extra twist to that. Just as it adds an extra layer of genetic variability, so it adds a separate or another layer to our understanding of how genes are inherited as a package or not as a package, as the case may be. So you remember I told you to remember the connection between red hair and freckles and green eyes. The fact that we know that those are inherited very frequently suggests that the genes for those may actually be located, or some of the genes involved in those may actually be located on the same chromosome, because they seem to be linked together. Certainly in the case of red hair and freckles, that is probably the situation. I don't know enough about it to say with absolute certainty, but certainly the evidence suggests that that is the case. So now we need to talk about the consequence of gender being the product of chromosomes, and the fact that the X and the Y chromosome in mammals, and certainly in humans, are of very different sizes, and that the Y chromosome is missing a large chunk of DNA that is present in the X chromosome. And that missing part of the Y chromosome that's present in the X chromosome does contain genes. And because a male only receives one copy of the X chromosome, and there is no alternative, whatever genes and alleles of those genes that happen to be present in that region of the X chromosome represents the alleles that the male is going to show. This is referred to as X-linked inheritance. So here is the gender determining situation as far as humans are concerned. And I introduced you to this in the previous presentation for the previous chapter, for chapter 8. But here we now need to talk about the consequences of this. So a male has 44 autosomes plus the gender chromosomes. A female has 44 autosomes plus her gender chromosomes. A male has the XY pair, a female has the XX pair. Who is responsible for determining gender? The male, because it's the male sperm that has the alternatives, the X versus the Y sperm. So if you are wondering why you continue to have children of the same gender, look to the father, because it's the father's sperm that are determining the gender of the children. It's got nothing to do with the mother. The mother can only provide an X chromosome. The father can provide an X chromosome or a Y chromosome. So he determines gender. However, when it comes to X-linked inheritance, it's all about the X chromosome that you get from your mother, not the X chromosome that you get from your father. Let me explain why. So if we take a look, and I've jumped over some slides, I know. Well, let me just draw the diagram in here. So if we have a look at the situation for a female, and if you're not familiar with this symbol, you ought to be. That's the symbol for female. And this is the symbol for male. So this is the 2 X chromosomes for the male, for the female. This is the XY for the male. Any genes that are located in this region the male has a matching region in the Y, so does the female. And in fact, for the female, she has matching regions all the way down, but the male is missing this region. So whatever genes happen to be located in here represent the genes that the male must express, because he's only got one copy. Therefore, that's what he's going to express. For the female, if the alleles are different, which they frequently are, then if one of these represents a mutation that could be potentially dangerous, there is probably an alternate version on the other X chromosome that is perfectly normal. And fortunately, most mutations are recessive. So for females, X-linked inheritance is not a huge issue. Is it possible for a female to show some of these conditions? Yeah. And red-green color blindness, as mentioned here, is one of those issues that it is possible for a female to actually have. And I've had a red-green colorblind student in a class a few years ago, and she actually got very excited when we started talking about this because she finally understood the inheritance pattern that gave rise to her red-green colorblindness. And I can show you that pattern on the next slide. So the big deal here is whatever alleles of the genes that are present in this region of the X chromosome are present, are the alleles that the male is going to express. And red-green color blindness happens to be one of them. So the gene for red-green color blindness is located in this region. The gene for hemophilia is located in this region. The gene for a type of muscular dystrophy called Duchenne's muscular dystrophy is located in this region as well. So if the X chromosome that the male receives from his mother, because he must get an X chromosome from his mother. Why? Because the Y chromosome he gets from his father. If he gets the Y chromosome from his father, he's male. If he gets the X chromosome from his father, he is now she. I mean, that's the thing that you need to remember. For a male to be male, he must get his Y chromosome from his father. His X chromosome he must get from his mother. So the X chromosome with the mutations in it originates from his mother. So this is important to recognize. The X chromosome that the male has always originates from his mother, not from his father, because he got the Y chromosome from his father. That's why he's a male, not a female. So let's take a look at the inheritance patterns. So if we take a look at the symbols, you'll notice that we put the X and the Y in here, and we put uppercase and lowercase letters against the X because that's where the mutation lies. We don't put it against the Y because there's no matching region in the Y that contains one of these genes. That's the reason why this is referred to as X-linked, because the genes are located in that region of the X that is not matched by the Y. So this is a normal female having offspring with a male who has the condition, is X little n, he's got the condition. This is referring to color blindness, so this is a colorblind male. So here are his sperm, here are the mother's eggs, and you can see that the females all get an X big N from their mother. They get the X little n from their father. They must in order for them to be female, but that makes them all carriers. So one of the things that we need to remember here, there is no male carrier in X-linked inheritance. He either has the condition or he doesn't. There's no intermediate. He cannot be a carrier. Females, on the other hand, can be carriers. And so here you have the daughters as carriers. Now we have the situation where the mother is a carrier and the father is perfectly normal, does not have color blindness. So all of the females receive an X big N from their father, but half of them receive an X little n from their mother. Remember, 50-50 chance. As far as the males are concerned, 50% of them get the X big N from their mother, but 50% of them get the X little n. That makes them colorblind. Again, remember, if he gets the X little n chromosome, he's got the condition, in this case, colorblindness. So this is the origin of the male, of a colorblind male. So under what conditions can a woman or a female have colorblindness? And this is the situation for my colorblind student. Her mother had a colorblind father, so her maternal grandfather was colorblind. And because he was colorblind, this would be his situation. Here's her grandfather. This is where her mother came from. So here is her mother, a carrier of colorblindness. Her father was himself colorblind. So my student's father was colorblind. And so under these circumstances, half of the females get the X little n from their father, but get the X big N from their mother. So they're carriers. But half of the females, and this is my colorblind student, gets the X little n from their mother and the X little n from their father. And so this individual is a colorblind female. This is my colorblind student. I didn't find out whether she had any brothers who were colorblind, but if she did, half of her brothers would potentially be colorblind and half of her brothers would be vision normal. Now, you'll notice that there's no cross for a colorblind female and a colorblind male. And the reason for that is all the children are going to be colorblind because everybody gets the X little n from all the daughters get the X little n from their father, and they would get an X little n from their mother if she is X little n, X little n. And all the guys are going to get an X little n from their mother. And so that means everybody's going to be colorblind. So if my colorblind student married a male who was colorblind, then all of their children would be colorblind. No exceptions. So you can see that these inheritance patterns are very, very different from anything that Mendel might have predicted. Because remember, in everything that we talked about as far as Mendelian genetics is concerned, gender did not even enter into the issue. There was no consideration of gender. But now gender becomes really important because the gender chromosomes do contain genes other than the ones that determine gender. And that means if there is a difference in the chromosomes, which there most certainly is in humans, genes that are present on the X chromosome that are not present on the Y chromosome represent the characteristics that the male will have. So as far as red-green color blindness is concerned, you can live with that. You can learn to survive by memorizing which position the red light versus the green light is in on the traffic lights and that sort of thing. There are some things that you can't do if you're colorblind, so you can never be a pilot if you were colorblind because they use red and green lights for markers on airplanes. You've probably seen that at the airport. And there are some other things that you can't do as someone with red-green color blindness. But hemophilia, that's a different situation because this is potentially lethal because your blood doesn't clot. And these individuals have to get constant inputs of the missing blood factors in order to allow their blood to clot. They need to go and receive a new input of the missing proteins about once every 3 or 4 weeks in order to be able to have blood that will clot instead of just bleeding continuously. And this originated, we think, with Queen Victoria, because it was the marriage of Queen Victoria's daughters into the royal families elsewhere in Europe that led to the appearance of hemophilia in the royal families of Europe, most notably into the Russian royal family. And so this is a picture of the last Tsar, his wife, and their kids. And this is Alexis, their only son and heir to the throne, and he was a hemophiliac. And in part, you could blame the first Russian Revolution, because there were two. The first Russian Revolution led to the downfall of the royal family. And you could blame some of that on the fact that the parents were so concerned about the health of their son that they got involved with trying to prevent their son from dying as a consequence of the hemophilia, and weren't paying a whole lot of attention to what was going on in the country. Not that they paid a whole lot of attention in the first place, but they paid even less when Alexis was born. And so they were removed in the first revolution. The second revolution that took place a few months later brought the Bolsheviks, the communists, to power, and would ultimately lead to the execution of the whole royal family. So the course of world history has been affected by hemophilia, the presence of a recessive allele, which is kind of an interesting thing to think about. So the final part of the chapter brings us back to thinking about dogs again and the history of dogs as a species, because we talk about Canis familiaris, that is the biological name for the domestic dog, and the origin of the dogs. And so people have spent some time looking at the genetics of dogs and doing comparisons between the genetics of dogs. And this is how we think the various breeds may be related to each other. So some of the very earliest breeds of dog are the Asian dogs. And the Basenji, I think that's Egyptian, if I remember correctly, but the Akita and the Shar-Pei. Akita is from Japan, Shar-Pei is obviously from China. Then we have more dogs that have an Asian association. Middle Eastern association, and then dogs that we associate more with Europe as later products of man's breeding of the dogs to give rise to it. This is one family tree for dogs. It's not necessarily the only family tree that we could draw for dogs, but this is one family tree for dogs. So I've got a few more genetics problems. This is the last two slides. I promise you these are the last two slides. And these are just some more genetics problems to make sure that you have clear in your mind some of the things that we've talked about. It's a lot of information to digest, I know. So, which of Mendel's laws are associated with the following events of meiosis? Because remember, we have to be able to recognize where in meiosis we see the behavior that leads to Mendel's two laws. So the random and independent arrangement of the chromosomes in metaphase 1. Well, as soon as we say random and independent, we think independent assortment. So that's the law of independent assortment, which means separation of homologous chromosomes must be the law of segregation. So those are the connections between Mendel's laws and the events in meiosis, something that you need to make sure that you have clear in your mind. Where do you look in meiosis to see the behavior of the chromosomes that leads to Mendel's laws? And here are my last two concept check questions for this chapter. Explain why linked genes disobey Mendel's law of independent assortment. Well, Mendel's law of independent assortment is based on the fact that the genes are located on separate pairs of homologous chromosomes that behave independently of each other. And that's why why you get the different combinations of alleles associated with those chromosomes. If the genes are linked, they are on the same chromosome. And because they're on the same chromosome, they tend to be inherited together, not independent or separate from each other. And so that's why they disobey Mendel's law of independent assortment. The white-eye mutation in fruit flies is located on the X chromosome. We've just been talking about that. If a white-eyed female fruit fly is mated with a red-eyed or wild type, that's the term that we give for the normal feature, male, what are the genotypes and phenotypes of the resulting offspring? Let's do the Punnett square. So the first thing we need is the genotype of the female. Well, we're told that she is white-eyed, so she must be homozygous for it. The male is normal, so he must be perfectly normal. So here we can draw our Punnett square. We, by convention, put the female down the side when we put the male across the top. So we've got X little n, X, X little n, X, X little n, Y, X little n, Y. And I would always encourage you, as you first start out with genetics problems, to draw out the Punnett squares until you can start visualizing them in your head, if that is possible for you. Otherwise, you're likely to make mistakes. So what have we got? Well, the XXs are obviously females. Being X little n X, they would be females and carriers. Let me erase this for a minute. Put it this way. Females are normal, but carriers. Males all have white eyes. So the females would be normal, they'd have red eyes, but they're carriers. And the males would all have white eyes. And again, notice why. The males are getting their X's from their mother, and she is X little n, X little n. So there are no alternatives. Every male gets the X little n. He gets the Y from his father. Remember, if he got the X, he would be she. That's the way it is. The females must get X's from both parents, so they get the X little n from their mother. Again, no alternatives. And the X from their father is a normal X. So they don't have the condition. They're not homozygous for it, but they are carriers for it. So that means they would be red-eyed, normal, but they would be carriers of the white eye mutation. So that's it. This has been a long chapter. I realize that. There's a lot of information here. But we've made it, and it's only taken us about 2.5 hours, I think, in total. No, it's probably longer than that. It's probably about 3.5 hours. And this is the reason why I really wish the authors of the book would separate this out into 2 chapters. It's a lot of information for a single chapter. Anyway, as always, if you have any questions, concerns, or comments about anything that I've covered here, by all means, come and see me in office hours. Otherwise, I will see you back here again for chapter 10, which is looking at DNA and its role as the molecule of inheritance. See you then. Bye for now.